Okay, so hopefully you guys are seeing this. And I realize I probably can't move that. Oh, that's weird. Can you guys still see the slides? Everything's a little funky. Why is this? I don't want that.
Yeah.
No. Okay. No, I can't because I have this big thing that says give subtitles a try. No, go away. Okay. All right. Whatever. Yeah, I don't want subtitles. All right. Anyway. Well, no subtitles, but that's the only way I can get rid of that. Okay. This is actually kind of nice, except for the weird subtitles down there. So this chapter talks about the biosynthesis of amino acids, nucleotides, and a whole bunch of related molecules in here. Um, so what I want to cover today is nitrogen fixation, and then we'll talk about ammonia incorporation and the biosynthesis of amino acids, and a little bit about heme. Okay, so that sounds like a lot to cover in one day. We're actually going to get through about 2/3 of the slides today, but we're not going to go into a lot of detail. Like the first quarter of the slide deck is just kind of listen, get an idea. We can, I'm thinking what I should have done is cut it down to like 3 slides instead of 20. So. But let's start with a little bit of background here. And we've sort of seen this with the urea cycle. We know how important it is to maintain nitrogen balance in the body, but nitrogen is immensely important in biochem. It's one of the most common elements in living organisms, and we find it in proteins and nucleic acids a tremendous amount. But it's also in pretty much every cofactor that we talk about, our NAD, FAD, because of course they have that nicotinamide, adenosine. So adenosine. A lot of hormones will have nitrogen in there. A lot of neurotransmitters, pigments, defense chemicals. All sorts of stuff has nitrogen, so it's everywhere. That's why we got to learn about it. If we look at nitrogen on Earth, we know our atmosphere is about 80% nitrogen, but that nitrogen gas is not very useful to us because it's inert. We cannot convert into ammonia very easily. We can do it, but it just takes a tremendous amount of input in here. So it's got a very high activation energy. If you look at that, that's about the same activation energy as our typical ATP. So like, oh, file that away for later. There are non-biological processes that can break this triple bond between the 2 nitrogen atoms. If we have a lightning strike, which I don't know if that storm yesterday, I didn't see much lightning, but supposedly there were some lightning storms around. We can actually combine nitrogen and oxygen through that massive electrical discharge to create nitric oxide. We also can combine nitrogen and hydrogen to make ammonia through this Haber process. And the only problem is, we don't exactly want to have lightning bolts striking living organisms, nor do we want living organisms to have super high temperature and super high pressures like the Haber process requires. So biologically, there have to be ways to incorporate or fix nitrogen into useful forms. Many organisms have developed methods to do this. Most of them are single-celled prokaryotes, various archaea, and some bacteria. We'll find a lot of bacteria that will live in symbiosis with plants. So we tend to think about bacteria as like, oh no, I got strep throat, or oh, I got an infection, or maybe, oh, I have bacteria in my gut that help things out. But we also see a lot of symbiosis with plants, where the bacteria will produce useful products that the plants can use, and the plants will kind of help those bacteria thrive as well too. We also see that some of these bacteria will live in symbiosis with animals. I don't know how much you think of termites as animals, but technically they're animals. And the reason that termites can digest wood, the cellulose in there, is because they have a bacteria in their saliva the bacteria digest the wood, and you can get useful products from that for the termite. So we have these various spirochetes in here. Apparently we're going with our British spelling today. But these various bacteria tend to have enzymes that can overcome that high activation energy, and they will utilize ATP to do that. Okay. So If we look at that, go back to that Haber process, and I think I've mentioned this like 2 or 3 times over the course of the year. It is one of the most significant developments by humankind. You can put that up there with, harnessing fire and inventing the wheel. Because with the Haber process, we can make fertilizer, and having fertilizer allows plants to grow, so that we can both consume plants and feed animals that we can consume as well too. So roughly, the artificially made fertilizer accounts for the nutrition value of about 1/3 of the human population. So about 2 billion people are fed because, well, more than that now, getting close to 2.5 billion people are fed due to industrial fertilizer. So that is a massive step forward. And as bad as things are in the world, if you look at the percentage of people who are at risk for starvation, it's at one of the lowest points in human history, simply because of this process in here. Unfortunately, this is energy intensive. I mean, it requires very, very high temperatures, very, very high pressures. So there are always, methods to try and improve this. One of the things that we have been looking at over the decades is mimicking biological fixation, so that we might get some energy savings there, or allow more renewable energy sources to do this. So let's talk about nitrogen and what happens here. So that's weird. We got this. these are in the wrong order. Well, I didn't catch that when I was prepping. Nitrogen, which is actually over here, is into, and it has that covalent triple bond. All right. We have our nitrite, which is this one. Oh, that's terrible. Which is NO2. It's light on oxygen. You can think of that ite for light on oxygen. It only has 2 oxygens, and nitrate has 3 oxygens. And it ate the other oxygen, I guess. I don't know. So basically, we go from a neutral nitrogen to a +3 nitrogen to a +5 nitrogen, and then in our ammonia, we actually go to a -3 nitrogen. So we can see our oxidation state changes there as well too. This slide is terrible. I can't believe this. I don't know how many years I've been using. Well, only a couple years I've been using the slide, but I've never noticed that before. But anyway, now that it's on a big screen in front of me, this is all wrong. Dear Lord. All right. Anyway, those are our 4 primary compounds: nitrogen gas, nitrite, nitrate, and ammonia. And they kind of go in that order. Okay. So we have a nitrogen cycle in here. And this is going to be a series of chemical transformations to give us a good balance between nitrogen gas and biologically useful forms of nitrogen. So we can have fixation, where bacteria will reduce nitrogen gas to ammonia. Okay. We can have nitrification, where we can oxidize the ammonia into nitrite or nitrate. Then we can have assimilation, where we can take this nitrite and nitrate, and convert it into ammonia through the action of a couple specific enzymes, nitrite reductase and nitrate reductase. Okay, so remember, we're gaining electrons, so our +3 or +5 ammonia is getting converted into nitrogen -3. Sorry, nitrogen +3 and +5. And this ammonia gets incorporated into amino acids and so on. Organisms die, and when they get ammonia returning to the soil, and then the bacteria can convert that ammonia back into nitrite and nitrate. We can also have denitrification, where nitrate is reduced to nitrogen under anaerobic conditions. Okay, and under anaerobic conditions, our nitrate becomes the ultimate electron acceptor instead of oxygen. So we've got a whole bunch of these processes in here. If there's anything that you need to know through these first slides, it would be this one. We've got fixation, nitrification, assimilation, and denitrification. And then just knowing that the last one is anaerobic, right? We can look at this visually as well too. Okay, so normally we will see, here's our atmospheric nitrogen. Typically it goes to ammonia, and then we can incorporate that into various things, or we can have our ammonia get converted to nitrite and then converted into nitrate, and our nitrite can get converted back into nitrogen. Okay, so up until about 10, 15 years ago, This was a circle with these two little legs off of it. We're going to talk about something weird that happens here in the middle, where you can go directly from ammonia to nitrogen. That is a unusual and relatively newly discovered process for this Anammox bacteria. But that's new and it's incredibly rare. Or incredibly unusual. All right. When we look at assimilation, so assimilation, we are, ultimately converting our nitrates and nitrites to ammonia. We have two key enzymes in here: nitrate reductase, which is going to take our nitrate and convert it into nitrite. Right. So we add 2 electrons, we lose an oxygen. This involves a cofactor that contains molybdenum. All right. So one of our rare metals in here that becomes critical. Molybdenum is important in this cofactor. This also involves NADH as well too. Just kind of pay attention to this because I'm not going to make you know that. You're not going to have to memorize any of this that we're talking about. We then have nitrite reductase, which can take that nitrite and then again reduce it and convert it into ammonia. This involves a ceraheme. We have this iron sulfur center in our ceraheme in here. We have a whole bunch of these electrons, and our electrons now are coming from NADPH instead of NADH. Okay. Again, none of this is happening in animals. We're talking about plants and bacteria. So, but we've now gone from nitrate to nitrite and nitrite to ammonia. All right, and then that ammonia can be used. Okay. On the other hand, we have nitrogen fixation, where we can take nitrogen gas and directly convert that to ammonia. This is very exergonic, very slow due to that triple bond's high activation energy in there. So we can release a lot of energy, but that you got to get over that hump there. That activation energy is very prohibitive. So there is something that some bacteria have called this nitrogenase complex that will use ATP to overcome that hump. And it takes about 16 ATPs to convert one nitrogen gas to 2 molecules of ammonia. So that is a very expensive process energetically, just to get over that hump. This nitrogenase complex is massive. It has a huge number of unusual metal ion centers in here. We have this FeMo cofactor, which involves iron and molybdenum. Some organisms will use vanadium instead. We're essentially, if you think of our electron transport chain, we are passing electrons through this huge number of metal centers, and that's providing the energy to fix nitrogen. All right. Again, don't need to memorize any of this. Our FeMo factor has a bunch of, it's fun to say FeMo, but we got iron, sulfur, molybdenum, and a homocitrate in there as well too. Massive electron flow through here. So we're just kind of, having our electrons move, and that's providing the energy, the current to have this process go. Okay. We'll also see hydrogen gas that gets formed as a side reaction of this as well too. But if we were to try and trace all this through here, we could take our pyruvate, to acetyl-CoA. We got ferredoxins, dinitrogen reductase, more of this reductase, this dinitrogenase down here, nitrogenase, whatever. And eventually, ultimately, after transferring a huge number of electrons, we get ammonia and hydrogen. All right. I just want you to be familiar with the process in here. There's a bunch of redox reactions that occur. Again, bacteria in here. How does this occur? We really aren't sure. We have 2 logical pathways for intermediates in here. We can have this nitrito-imido pathway, or we can have this hydrozido pathway as well too. Like hydrozido, what the hell is that? That's where we have this, these functions over here. So either we can look at this kind of triple bond to the metal center, or a kind of a hydrazine attached to the metal center. Either way will work. Don't care. Don't need to memorize it. Just know that we have this complex procedure for fixation, for converting nitrogen to ammonia. Okay. What we typically see is bacteria are doing this, and they're living in a symbiotic relationship with plants or legumes, fungi, all sorts of stuff like that. I think we have a picture coming up of pea roots where we'll actually kind of false color in so you can see the bacterial symbiotic bodies that are in there. The important thing is the bacteria have access to the plant's carbohydrate and citric acid cycle intermediates. So the bacteria get energy from the plants, and then the bacteria provide ammonia for the plants. They'll often provide more ammonia than the plant needs, so the excess can be released to the soil. And again, this is one of the ways, if you think of agriculture, mulch, often will like treat your garden with mulch or something like that. Plowing under various plant life will allow it to decay and provide ammonia to the field so that there will be sufficient ammonia reserves available for new plants to grow. If we kind of look at, I forget what flower this is, but these are the root nodules in here. Bacteria are in these root nodules and allow the production of ammonia so that you go through the roots up to the plant and allow it to grow. Here we have a false color electron micrograph. Everything in red are bacterioids. These are the things that are providing the ammonia that allow the plant in the kind of the blue, yellow, and green to grow. All right. So to kind of recap everything that we've been talking about here, we've got two different terminologies: assimilation versus fixation. Assimilation is where we take nitrate or nitrite and convert that to ammonia. And fixation, we go directly from nitrogen gas to ammonia. Okay. Assimilation uses NADH and NADPH and a whole bunch of electron transfer in there, whereas fixation uses a ton of ATP. Right. Both of them are electron transfer processes. Both of them have molybdenum in there and multiple redox cofactors and stuff like that. So if you were to get a question about any of this stuff we've been talking about through these first 20 odd slides, it would be probably on, what's the difference between assimilation and fixation? All right. I mentioned there was a weird, relatively newly discovered process, this ANAMOX, that stands for anaerobic ammonia oxidation. And this is where some bacteria can directly oxidize ammonia and nitrite into nitrogen gas. So it kind of short circuits the nitrogen cycle. There's no denitrification step. This could be used in waste treatment for cheap ammonia removal. So if we have, excess ammonia, we can get rid of it. Why is this weird? Because these bacteria rely on an enzyme called hydrazine hydrolase, where they are converting ammonia in this ammonia hydroxide compound in here into something called hydrazine. Okay, and hydrazine is rocket fuel. That's literally, we use that for rocket fuel before we discovered that bacteria make it. It's also incredibly toxic and very damaging, but there's this hydrazine oxidizing enzyme within these bacteria that will give us this nitrate reducing enzyme, so on and so forth, and you can actually get nitrogen gas out of this. The interesting thing is the bacteria will produce, so as you get to nitrogen gas, you have these 4 protons and these 4 electrons that get passed up to this enzyme in here. These 4 protons can go through their ATP synthase and be used to produce ATP. So it's actually a really neat way to make ATP for the bacteria, but they're using rocket fuel, this very, very toxic substance that would kill most organisms. How do these bacteria do that? Okay, they have some, really unusual structures that allow them to metabolize hydrazine. Okay, so hydrazine would normally diffuse across membranes. Okay, so to prevent that, these bacteria have phospholipids that are made up of laterales. Okay, and what the hell is a laterale? Well, a picture helps. They have this structure in here like this, and these ring structures in here that are attached to the lipid chains actually block the diffusion of hydrazine across the membrane. So here's our normal phospholipid molecule. We can kind of put our hand over the yellow part there and you wouldn't see that. But because of these laterales that are in there, they block that diffusion across there. So that's what allows the hydrazine to accumulate within the hydrosomes and be metabolized. Let's see. I think there was something else here as well too. Nope. Okay. All right. So that's the weird process in there. And if you go back to that circular picture of the nitrogen cycle with the legs, and then this amomox is in the center there. All right. So that is approximately 1/4 of this topic. And all I want you to know, assimilation versus fixation. You have your nitrogen gas, your nitrate, nitrite, and ammonia. Those are our 4 important nitrogen compounds. That's it. Not too bad, right? Because I know this chapter is awful in a lot of ways. Okay. So anyway, because I want to be anthropogenic and talk about humans and animals and stuff like that, we just want the ammonia. And how do we, once we get ammonia, no matter how these bacteria and plants make it or whatever, now we have ammonia, we can incorporate ammonia into biomolecules. So now we can actually talk about what we're doing here. And typically, just like we saw in the urea cycle, glutamate and glutamine are our, temporary reservoirs of excess ammonia. Glutamine will kind of store excess ammonia and get it to the liver, and then you can start pushing it into the urea cycle. So glutamine gets made from glutamate by this glutamine synthetase in a 2-step process, right? So here's our glutamate. We have our lovely little carboxylic acid side chain. First step, we phosphorylate that, right? And then phosphate makes a good leaving group, so we can get rid of the phosphate as we add our ammonia, and now we have glutamine. Okay, seen this before? We're all good there. All right, this is what our glutamine synthetase looks like. Here are our binding spots for our ATP, ADP, and our glutamate. All right, so just to poke a little bit at the regulation of this, we can adenylate this tyrosine 397, which is not shown in here, but if we add an adenine group to that, it will actually increase sensitivity of our glutamine synthetase to various inhibitors, which will slow down the production of glutamine. Okay, so this is just another way of regulating it. Okay, we also have this regulatory protein, which we'll call P2 or PII, or something in there, which we can see there's this whole cascade of stuff that involves, either adenylation or deadenylation. So when we deadenylate our glutamine synthetase, it's active. When our P2 is activated, then we will tend to adenylate the glutamine synthetase and inactivate it. On the other hand, if we uridylate P2, then that will actually deadenylate the glutamine synthetase. Oof. And of course, we can see our glutamine. We can have this, feedback. This can function as an inhibitory molecule to our uridylate transferase, which will prevent the uridylation of P2 and promote the inactivation. If we have lots of glutamine, we don't want to make more. If we have high levels of ATP, we'll see the opposite direction here, and so on and so forth. I don't want to go into massive detail about this because I'm really, this is a model mechanism, but it's not something that I'm going to quiz you on in detail. Okay, so glutamine synthetase is regulated multiple ways, though, because it is so important to our initial nitrogen balance within the body. Okay. All right. There's another key factor here. Our uridylated P2 upregulates transcription of glutamine synthetase. So if we go back here, uridylated P2 wants to activate this. That indicates we need to make more glutamine. So not only will it bind to the enzyme and trigger this active form of glutamine synthetase, it can also go to the nucleus and activate transcription of glutamine synthetase, so we have more capacity to make more glutamine. All right. So there are multiple levels of regulation because, again, our nitrogen content in our bodies is of critical importance to living organisms. So that's why we see so many regulatory factors in here. You might get a question about that. Why are there so many ways of regulating this, or describe multiple ways or something like that? Okay, so let's pause and take a breath. To make our amino acids, we need to incorporate ammonia, and there's a ton of ways to do that. We're often going to start from glutamine or glutamate, but there are a whole lot of these aminotransferases, and they do just what they say. They transfer an amino group from one molecule to another. We saw pyridoxal phosphate in our previous exam. We're going to see it again here. Pyridoxal phosphate is a very common cofactor for these reactions as well too. So just like we saw that pyridoxal phosphate, being important for transferring ammonia and getting it into the urea cycle, make our carbamoyl phosphate and eliminating urea. Going the opposite way, the same thing is going to happen. It's just going to be, instead of transferring this way, we're going to transfer the other way. We took the bus into the city. Now we're going to take the bus back out of the city, I guess. I don't know. There are mechanisms. Be aware that there are mechanisms. All we're concerned about aminotransferases in here. So, how does this happen? All right, there are seven common starting materials for all of the amino acids. Okay, bacteria can make all 20 amino acids that we incorporate into proteins. We can't. We have to get some in the diet. Okay, but depending on what amino acid we want to make, we need one of these 7 compounds: ribose 5-phosphate, erythrose 4-phosphate, 3-phosphoglycerate, phosphoenol pyruvate, or alpha-ketoglutarate and oxaloacetate. Okay, so we can see here erythrose 4-phosphate, we can make tryptophan, phenylalanine, and tyrosine. Okay, ribose 5-phosphate, we can make histidine. That's a weird one, it only makes one. Same with phosphoglycerate, we'll make serine, but serine can be used to make glycine or cysteine, so we can really get 3 for the price of 1 there. Phosphoenol pyruvate, much like erythrose 4-phosphate, can be used as a precursor. You can see what we have in common here: tryptophan, phenylalanine, tyrosine. They all have ring structures, right? Okay, pyruvate, alanine, valine, leucine, isoleucine. Alpha-ketoglutarate to glutamate, we've seen that as a kind of a transfer mechanism before, but now we can make glutamine, proline, and arginine from glutamate. Oxaloacetate to aspartate, and then asparagine, methionine, threonine, and lysine. Do you need to memorize these? I don't think so. I don't think I'm actually going to have you guys, like I say, I think exam 3 is going to be all take home. All right. Okay. So where are we going with this? This is our overview. This would be, was it slide number 32? So we're a third of the way through the chapter. This is a good one to focus on. Okay. So all amino acids derive from one of those 7 precursors. Okay. And these precursors can be either part of the citric acid cycle, part of glycolysis, or part of the pentose phosphate pathway. We go back here. Here's our citric acid cycle. Here's our glycolysis steps. And, here's our pentose phosphate pathway. Okay. All right. So that's where they're all coming from. And it's just how we rearrange them in there. Just another table kind of showing what we can make from them. I like this picture better because I think it's a little easier to see, but if you like tables better, this is a good thing to look at. All right. So I'm briefly going to go through these, but I don't want to talk about these in any huge detail because, like I say, I want you to be able to utilize the information, not necessarily just memorize it. Okay. Which is why I'm going to make the exam take home. So application. Okay, so alpha-ketoglutarate, if we go back to chapter 18, we've seen that in our urea cycle in there. We can convert that into glutamate, and then we can use glutamate to make glutamine through our glutamine synthetase, or we can use other enzymes to make either proline or arginine from it. Okay. If we look at that for the proline and arginine, glutamate, you can make this glutamal phosphate, this glutamate semialdehyde, this purine carboxylate, blah, 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 and then we get proline from it. Okay. So multiple steps in here. We don't really need that. For arginine, it's a little bit simpler. We see this ornithine delta aminotransferase in there. So, we start into the urea cycle to make ornithine, right? Remember ornithine from the urea cycle? But instead of continuing on the urea cycle, another enzyme acts on it to give us, our stuff to get the proline in there, okay? Ornithine, our citrulline to arginine, arginosuccinate to arginine. Sorry, I think I misspoke there, okay? If we go into the glycolytic pathway, we can get serine from this 3-phosphoglycerate. A couple steps in there. Oh, we've got serine. Well, if we've got serine, we can, cleave off this this group here to make glycine. All right, yay, good times. All right, not going to sweat it. We can also make cysteine from that as well too. So now we get, instead of getting rid of the whole CH2OH group, we just get rid of the OH group and replace it with a sulfhydryl group, okay, to make cysteine. Okay. All right. This is the mechanism for bacteria and plants. Animals have a slightly different mechanism to accomplish the same thing. Not going to sweat it. Okay. Oxaloacetate, our citric acid one, we can convert that into aspartate. Okay. I think we saw aspartate into oxaloacetate. Now we can go the reverse direction, oxaloacetate and aspartate. And then our aspartate can be acted on by various enzymes to make these compounds. And pyruvate, we can make these compounds. And notice that threonine can get converted into isoleucine. So there's a couple different ways we can make isoleucine. Okay. So we often have multiple pathways to make different amino acids. And that's good because that allows us to kind of fine tune the different quantities that we want. Okay. All right. Pyruvate in there, same way. Our aromatic ring structures, we get a little funky in there. We got a lot more complicated chemistry. Good thing we're not going into excruciating detail about these mechanisms. We have to make the ring structures. So we're starting with little linear structures, but we got to make these ring structures in here. So we're going to see this is where erythrose 4-phosphate comes in. But one of our common intermediates for all these is going to be this charismate. So whether we're making phenylalanine, tyrosine, or tryptophan, notice there's 2 ways to make tyrosine out of this. We're going to make this charismate, okay? So look, we got 7 steps, starting from phosphoenolpyruvate and erythrose 4-phosphate to get to this charismate. Okay, once we have this charismate, it's so charming, haha, little word play on charisma, that we can do other stuff in here. And again, if we go to histidine, what we have here, a 9-step process from ribose 5-phosphate to histidine. So if our pentose phosphate pathway, instead of making our nucleic acids, we can make histidine instead. All right. We're not going to sweat that. Okay, what I do want you to focus on is how do we regulate what amino acids we're making? Okay, so we can make all these different amino acids, right? How do we regulate them? And there's a couple different ways. One of the most common ways, and I think we've seen this feedback inhibition as an example of like isoleucine feeding back at the beginning. Oh, we got lots of isoleucine, let's feed back to the initial enzyme in that pathway and slow it down. And that's what we'll see. Our initial enzymes in there are often the targets. Our ultimate product can bind to that initial enzyme and slow it down. Okay. So feedback inhibition is nice and easy. Isozymes are another way we can do this. Okay. So isozymes, if you think of, maybe you've got one pathway that can lead ultimately to 3 different amino acids. A little picture there. Yeah, 3 amino acids. All right. We can use different isozymes that have different specificities. Okay. Sorry. So here's our example of feedback inhibition. Isoleucine feeds back and binds to this threonine dehydratase. All right. Isozymes. If we look at aspartate, aspartate can make either lysine, methionine, threonine, or isoleucine. So we have different isozymes. There are 3 isozymes in this initial enzyme. which are known as A1, A2, and A3. So our first step of converting aspartate to aspartyl B phosphate, we've got an A1 isozyme, an A2 isozyme, and an A3 isozyme. If we look at this, lysine will bind to the A1 and inhibit it, so we won't make as much lysine, okay? But we can still go through this pathway to make homoserine and methionine and threonine, okay? We can still go through this pathway as well too. Go down here, isoleucine, it will bind to A1 as well, all right, and inhibit that isozyme, but the other two function normally, okay? So let's suppose we go down to A2, we get down here, we have our homoserine to methionine and threonine. Well, methionine can come back here and bind to this isozyme at homoserine and prevent it from making more. Same way threonine can come back and bind to this isozyme and prevent it from making more, okay? Threonine will also go back and bind to B2. Isoleucine can also go back to B2 as well too, since if you make threonine, you can get to isoleucine. Okay, so isoleucine binding multipliers there too. If we look at this threonine to isoleucine, we have two isozymes there, C1 and C2, and isoleucine can feed back to C1, and inhibit that pathway. Okay, so if we have the C1 isozyme, we're going to inhibit it a lot faster than if we have the C2 for isoleucine. So we basically have slightly different flavors of the enzymes. The enzymes are all catalyzing the same reaction, but some of the enzymes are more sensitive to some of the end products. All right. And these isozymes become critical if we need more isoleucine and less lysine, then we want to stimulate these pathways going this way. All right, and block off this pathway over here. On the other hand, if we want more lysine, we want to block off these pathways and have it go through here. All right, does that make sense a little bit? The isozyme idea, I hope. No, shaking your head, no. Enzymes that catalyze the same reaction, but they have different conformations so that different end products can bind to them and inhibit them. All right. And it's, let's see here. I've tried to come up with a nice analogy for this, but like the game Plinko, or those carnival games where you like drop something in at the top and it slides down there all these different pegs and it can slide in different holes. You're getting like the tiny little center one, you win $1,000, and the other ones you win like nothing. I don't know. Yeah, okay. So that's essentially what we have here. We have all these different pathways that we can get down here to the bottom and make these products, but some of these isozymes, it's like putting a little bar up there. So, we've got to shunt it over this way. So we're kind of closing off this pathway when we make lots of lysine or lots of isoleucine. So we're going to shunt this to make sure it's more likely we're going to make something down here. On the other hand, if we have lots of threonine, we'll come back here and shut down this pathway and put a bar over here. So we're going to, be more likely to make something on the other side of the pathway. Hopefully that helps a little bit. I don't know. How do we make amino acids? It's like Plinko. Sorry, when I would skip school as an undergrad, I would hang out with one of my friends in town and we'd watch The Price is Right. Why? I don't know. That was in high school. Okay. So 7 different things we make our amino acids from, lots of different pathways that are regulated by feedback inhibition and isozymes. Okay. So once we make our amino acids, we can, of course, incorporate them into protein strands, growing proteins. Yay, that's all well and good. But we can also use those amino acids as precursors to make other things, like our porphyrin rings, various hemes, phosphocreatine, glutathione, various neurotransmitters, a lot of signaling molecules, cell wall constituents, all sorts of stuff. We can use our amino acids to make from. So again, going all the way back to the beginning of the chapter, we need nitrogen to make our amino acids. Now we can need our amino acids not only for proteins, but to make these other compounds. Okay. So if we look at porphyrins, we make our heme of the hemoglobin, our cytochromes, our mitoglobins. Porphyrin arises from reacting glycine with succinyl-CoA. Okay. For plants and bacteria, instead of glycine, it's glutamate. But this pathway gives us this delta amino levulinate, all right, levinolate. Well, yeah, I don't know. And this porphyrin. You don't need to memorize any of this stuff in here. But we can see, animals, succinyl CoA and glycine, and we ultimately get here, plants, glutamate, and we ultimately get here. All right. All right. Notice that we're using some tRNAs in here as well too to transfer our glutamate. All right. Yeah, we're not going to worry about that. Okay. Once we have this delta amino levinylate, then we can start making ring structures in here. So just like with our cholesterol, we start with our activated isoprenes and we combine, 5 or 7 or 10 or 20 or 30 of these units together to make whatever we're, to make our cholesterol. We start combining these small units together to make components. And then we can start assembling those components. I don't know, we should maybe name these after Avengers so we can say Avengers assemble, I don't know, whatever. And then the final step is we add our metal ion in there that gets coordinated. And we have our heme. Okay? There's all sorts of enzymes for all of this in here. All I want you to know is that, we can synthesize heme from our amino acids, particularly glycine and succinyl CoA to start. Okay. Most animals make their own heme, but there are lots of defects that are possible in this pathway. So mutations, misregulation can lead to various porphyrias. You guys like vampires? Vampire stories? Yeah. I had my son watch Lost Boys with me a couple weeks ago. Classic 80s movie. If you can get past the 80s fashion, it's still a really good movie. But vampires and the whole idea of vampirism may have stemmed from some porphyrias in here. Various porphyrias can, because they affect red blood cells, people would have a taste for blood. They kind of, want to, drink blood or bite blood in here. They often have very pale skin. Sunlight and skin would be very sensitive to UV light, so they avoid the sun. So all these kind of myths of vampires may have arisen from various porphyrias in there. So if we see this uroporphyrogen 1 that accumulates, it's really kind of neat because the urine can become discolored. So, if you have this particular porphyria, your urine might initially look normal, but then as it sits over time, it'll turn to pink or very dark purple, especially when you expose it to UV light or heat. So that's a good test for that. I hope to have a case study about some of these porphyrias for next week for you all. Teeth can also kind of show this red fluorescence under UV light. So you get like blood-stained teeth, if your fangs come out there, craving for heme because you're not making enough heme in there. So this is where we get our vampires from. All right. I'm thinking of a vampire question for the take-home exam, but I have enough crazy questions on there. All right, this is the one that I wanted to get to today to make sure I got to this. What slide are we on here? Oh, good, we're close. We're cool. Yes. Oh, we're gonna be fine. We're gonna be fine here. Heme. From heme, we can get various bile pigments in here. So heme, we can degrade our erythrocytes, and we have a couple different steps that we can make. The first thing we're going to take our heme and convert it into biliverdin, okay, which is this kind of greenish compound, which is what you see in bruises. So my leg now, my entire thigh is a combination of dark reddish purple from the pooled blood that has come to the surface. There's a fair amount of green as those erythrocytes are being converted into biliverdin. And there's a bit of yellow in the bruise as well too, as that biliverdin gets converted into bilirubin, although there's not much yellowish in there, and the yellow is more from the green kind of fading away, because the biliverdin, when it converts into bilirubin, gets taken away to the bloodstream and can be released in the urine in there. Okay, you make urobilin and stercobilin. All right. So these are our lovely blood compounds in here. So blood being red for the oxygenation, blue if it's deoxygenated, biliverdin for the green, bilirubin for the yellow. Okay. So here's what we see how this process works. Break this down into biliverdin. You see our biliverdin reductase, we get our bilirubin, and then we can transport that and make these different compounds in here. All right. So jaundice. When you have kids, they will test them for jaundice. And jaundice tends to indicate a lack of liver function because you tend to accumulate um, bilirubin in the skin, in the whites of the eyes. So you have kind of yellowish eyes or yellowish skin. This can happen due to impaired liver, maybe hepatitis or liver cancer, blocked bile secretion, gallstones, pancreatic cancer. Terrible, terrible things. Or if we don't have this glucuronal bilirubin transferase to process the bilirubin, we often see this in infants. So when kids are born, they'll check them to see if if they're jaundiced, because maybe their livers haven't started producing this bilirubin transferase in sufficient quantity, and they can treat with UV light. So you may see like newborn babies are taken to a room where they have a UV crib in there. Or we could just put them in sunlight as well too. And the UV light helps break down bilirubin so that you can get rid of it and won't accumulate it and won't develop jaundice. So this always leads me to a fun story about when my son was born. Yeah, you guys see me, Dr. White guy here, but my wife is Asian. So when my son was born, the doctor's like, well, he looks a little yellow. We want to test for jaundice. And I was like, well, he's half Asian. He better look a little yellow. So of course he didn't have jaundice. He just, was Asian. So always one of my fun stories there for my kids. And usually, a lot of children, won't have that glucuronal bilirubin transferase in sufficient quantities right after they're born, but usually within a couple days, that kind of comes online and they're producing enough in there. Right. So, I know early on we mentioned, creatine and phosphocreatine are very high energy compounds. They're more high energy than ATP. It's like, where do you get creatine? We can go to GNC and get a big bottle of it and get huge, man. Okay, no. You actually have to make that creatine and phosphocreatine somewhere, and it can be generated in the body by combining glycine and arginine, and then using this ADOMET. Remember, ADOMET is a methyl donor, a really specific, really good methyl donor. And then we can make our phosphocreatine that way. Okay. All right. Here we are breaking it down. And here we are synthesizing it, arginine and glycine. Combine those two, you get this guanidinoacetate. Oh, that's a wonderful name, right? Our methionine to give us our ADOMET in here. And gives us our creatine. And then we can phosphorylate this with creatine kinase to make our phosphocreatine. Okay. So that's where it comes from. All right, glutathione, very important for scavenging our oxygen radicals. So if we want to avoid any radical damage, we need our glutathione. It is present in most cells at very high amounts for that as an antioxidant reducing agent. So it helps protect the proteins and the metal cations that are being reduced, keeps the redox enzymes in a reduced state, and removes these various peroxides. We can oxidize this to a dimer using a disulfide bond. So we get this, glutathione dimer in here, GSSG is how I write that, as opposed to GSH. We actually have a picture of it here. Yes, glutathione reduced. Here is our reduced form. If we oxidize it to a dimer, basically this sulfhydryl group will combine with the sulfhydryl group of another glutathione to give us the cysteine bridge there. All righty. D-amino acids in bacteria. Now, almost all of our amino acids are L form. All the ones that are incorporated into protein are L form, but we can make these D-amino acids, and they arise from racemases in here. So bacterial peptidoglycans can contain this D-alanine and D-glutamine in there. Racemases can act on the D-amino acids and use pyridoxal phosphate as a cofactor to convert them back into the L form. Right? And racemase inhibitors are being used or studied as an antibiotic target. So because these bacterial peptidoglycans need D-amino acids, if we can inhibit these racemases, since we don't have racemases, we aren't making D-amino acids, only bacteria are, and bacteria need them to make these peptidoglycans, and our peptidoglycans are going to be important for the binding. If we can inhibit racemases, then maybe we can prevent these peptidoglycans from being made, which will reduce the infectivity of our various bacteria. Okay. All right. One example of that is this L-fluoroalanine is incorporated in there, and this cycloserine as well too. So cycloserine for tuberculosis and L-fluoroalanine as an antibiotic. Okay. Other things that we can make, plant lignans, hormones, natural products in here, spices like nutmeg, vanilla, like a vanilla candle. Oh, we've added, this vanilla scent in there. What is it? Cinnamaldehyde. Actually, one of the first labs I ever taught, I had a student who was excused from lab because we were working with cinnamaldehyde, and she had a very severe allergy to cinnamons in there. But other alkaloids like morphine as well too. Nutmeg, growth hormones for plants, these various polymers in the air. All of these can be synthesized from amino acids. All right. And again, here's our cinnamate in there. So, neurotransmitters, things that we've talked about before, epinephrine, so your dopamine, norepinephrine, epinephrine, these all can come from amino acids. Our tyrosine in here, like our L-dopa to dopamine to norepinephrine to epinephrine. Glutamate is a precursor for our gamma aminobutyrate, our GABA. This is how I know I have kids of a certain age, because I see that and I think, yo, GABA GABA. I don't know if you guys remember that, which was funny because, as cheesy as that kids show was, there were some good little beats and songs in there. Anyway, histidine to make our histamines. I know when I was first learning amino acids, I'd always confuse histidine with the word histamine, but as it's springtime now, some of you may be taking antihistamines for the allergic reactions in here, but we can make our histamines from histidine. Our tryptophan, we can use that to make serotonin. So all these incredibly important neurotransmitters are made from amino acids as well too. Okay. Also, nitric oxide, NO, is made from arginine. So I love this mid-80s discovery that nitric oxide plays an important role in blood pressure regulation, blood clotting, and so on. And you can synthesize this from arginine. There's this nitric oxide synthase. I will try not to talk about this way too much, but I have a fascinating story. So this was in one of my more, I think this was the advanced amino acids, advanced protein class that I had in grad school. We had a guest lecturer who was a friend of my PI, who was the chair of the department, who I think I've mentioned before was this very stern, seemingly overbearing, very intimidating woman. She brought in one of her good friends who worked in industry to talk about nitric oxide synthase. So who was also an older woman, but not nearly as more like Dr. Ruth, and smaller woman there, but very enthusiastic about her talk with nitric oxide synthase. We go back to this time frame and trying to regulate blood pressure, especially particularly people who had high blood pressure. This woman worked at a pharmaceutical company that was trying to use nitric oxide, or drugs that could improve nitric oxide to help regulate high blood pressure. They weren't having much luck, but they were in clinical trials. And one of the things they noticed in clinical trials, and this is one of the issues with clinical trials, is many clinical trials, particularly in the past, it's become more egalitarian now, but men were often used much more often than women for clinical trials. One of the side effects of this medication that they were trying to regulate blood pressure with for these men, the blood pressure wasn't super well controlled, but a lot of men reported getting erections. All right. Men, over and over again, they get the same side effect in here. So although they were never able to make a drug that, actually, I don't think they may have, but for blood pressure regulation, through serendipity, they invented Viagra. So there's your $1 billion story right there. So there's nothing like sitting in class listening to a discussion of nitric oxide synthase, and from this very nice, very old woman talking about erections all the time. And what I found more fascinating was the way that nitric oxide works. It diffuses. So it's not like you can actually, give people nitric oxide because it's a gas. So you have to give something, maybe an arginine derivative. And this would be a fascinating to look at, like the chemical structure of Viagra, so that you're actually inducing the promotion of the release of nitric oxide so that it can diffuse to where it needs to go within the body. And it is a kind of a localized effect, because you're just releasing nitric oxide and having it affect blood pressure within a small region, which I suspect, because we're trying to use this gas that can diffuse across membranes, is why it was so difficult to actually make a whole system blood pressure regulation in there. All right. Anyway, nitric oxide synthase is a fascinating molecule as well too, but that's way beyond what we need to know here. If anything, I would say that nitric oxide is, knowing that it's a gas would be what I'd want you to take away from this. All right. How are you guys doing? Yeah. It's like, we're still alive. That's good. I will say that right there. Okay. I actually apparently got too excited about doing this because I'm not sure I want to go any further with nuclear. Let me peek ahead here. Yeah, let's put this off until next week, because the biosynthesis of nucleotides is our next step. So the first quarter or third of this chapter is all background material, where the big takeaway is, nitrogen gas to nitrite to nitrate to ammonia, assimilation versus fixation. It's all background. And this is all done by bacteria and some plants for the most part. All right. Then once we actually have our ammonia, we get into the middle of the chapter where we talk about making amino acids. And these are all aminotransferases. And these aminotransferases act on one of 7 different precursors to make our 20 amino acids. Okay. I'm not going to ask you about mechanisms. When you get to take home stuff, I may be very specific, but probably besides these aminotransferases that are working on them, regulation of how we make them is important. We often have feedback inhibition, but if you can kind of study and wrap your minds around those different isozymes, because again, our aspartate, we can make 4 different products from that. Well, we're going to have different isozymes in those pathways to help limit that. Besides making amino acids, we can also make other important biochemicals like our porphyrins, or neurotransmitters, or stuff like that. Okay, so that's our beginning and our middle. The end of the chapter is all about nucleotide biosynthesis. And if we go all the way back to the beginning of the semester, we talked about glycolysis, and we kind of said, oh, we've got the pentose phosphate pathway. Here we're going to see our pentose phosphate pathway, where we have our ribose 5-phosphate as our precursor, and our ribonucleic acids, or our deoxyribonucleic acids. That's where our ribose is coming from. So here we finally get to that pentose phosphate pathway. We're going to see all these various nucleotides that we can make. So of course, we're going to talk about A, G, T, and C, and also U. And then there are other compounds that we can make from our nucleotides as well too. So we think of our nucleotides as being incorporated in DNA and RNA, but we're going to have our other biomolecules that we can use, like a ATP or GTP, or uridylation of stuff, or our CoA, coadenyl. Many compounds are adenylated, but we can also use these as precursors to make other drugs or other bioactive materials as well. Okay, so block out a nice long section of quiet time to kind of sit and study through this. What I want to do, right? Oh, did I do? Let's stop sharing here. Gotta figure out. Okay. Yep. Great. There's where that's the thing I wanted to turn that off from. And let me share something else. What is up here? Okay. So if we go into here, there should be some decent supplemental stuff to help out with this for the exam 3 material. And I will open up the rest of the homeworks here real soon as well. Fatty acid synthesis. So come back here. Our brief amino acid synthesis video with some trance music in there to put you in a relaxing, calm state of mind. You can see in the next chapter, we're going to get into diabetes and insulin glucagon and all that stuff in there. I don't know why I have these in here this way. These are, and some old Zoom stuff in there. Huh, that's weird. I have to look at that. But see how well you like this, because I know Lehninger can be kind of a pain in the butt because it goes into excruciating detail about every step, and not even every step, because some of those it's like, oh, we got 5 steps in between here and here, and we're not going to worry about those. I don't want to worry about the pathways or the steps in there. Maybe a few key intermediates or cofactors, but mainly regulation. We're going to talk about a lot about regulation in here, because you can always look up, well, how do I make serine again? What do I need to start from that? What enzymes are involved? I just want you to know, that feedback inhibition, the isozymes, transcriptional control, stuff like that. Okay. All right. But yeah, I think this is a good video in here, and not horribly long. I think I've gotten rid of the things that don't work. Sadly, I miss some of my good stuff. I'll see if I can find some other things in there for the nucleotide synthesis as well too. Okay. All right. So that's me rambling on way too fast, way too much, right? You guys overwhelmed? Yeah. But just block off some time, sit down, draw pictures if it helps, whatever strategy kind of helps in there. The
quiz is on 26 and 27, right? Okay.
What's that? I'm sorry, what was the question?
Oh, my bad. I did not unmute myself. That wasn't for you, Dr. Ebert.
Okay. Yeah. So, but yeah, this next exam is just chapters 21, 22, and 23. But 21 and 22 are long. 23 is not as long, but it brings everything together. So, where we are now is, all the stuff that we're learning. Next week, we're going to start tying it all together, and it can get crazy pretty fast. So we'll do some in-class work for that. I want to look at my assignments here real quick, because where did they go? I had to look at I had some good case studies and some good assignments, but then they kind of got crappy. Oxidation. All right. Well, anyway, but I'll find something that we can kind of discuss how this stuff works in real world environments in here. There are some very fun, well, maybe that's the wrong adjective. There's some interesting disease states that arise from defects in these pathways, right? Like the whole vampire thing there too. Okay, but aside from that, I'll stick around for questions. But this was a good hour and 10 minutes of intensive nitrogen discussion. So